Hello, everybody. Hi. Why don't we go ahead and get started? Welcome, everybody. I'm standing in for Dr. Rockbeisel today, who's at a hospital leadership retreat. But we're really excited to have one of our bright young new faculty come and talk today. So Dr. Jackie Klaus is going to be presenting. She is a child adolescent psychiatrist and a psychiatric neuroscientist. Jackie specializes in the care of adolescents and young adults with early psychosis. She attended undergraduate at Johns Hopkins University and completed her MD and her PhD at Vanderbilt University. After her MD and PhD, she completed adult and child and adolescent psychiatry training at Massachusetts General and McLean Hospitals, where she later joined the faculty. In 2024, she joined us here at the University of Maryland Department of Psychiatry in the MPRC. And in her current roles, Dr. Klaus provides clinical care, teaches, and supervises trainees. And she also conducts research on the neurobiological basis of risk and resilience to psychiatric disorders. Today, we get to hear some about that. So thank you very much.
Thanks. And thank you to all for persisting up to the third floor. I've been told that was a challenge. All right. Let's see, I'm going to move the chat and make sure I can monitor it as we go along. All right. Well, I'm excited to talk with you guys here today. I made the trek all the way over from Catonsville to join you. So I realize we sometimes are a little isolated out there. So I'm going to talk with you today about some work primarily that was done during my time at Mass General, but also some newer work that we have just started at MPRC related to psychosis risk and kind of dimensional psychopathology in that patient population. Is this working? I jinxed myself with the clicker by saying that I thought it was working. Okay. Okay. See if I can click it. Yeah. Okay, now we're working. All right. These are my disclosures, but it's not related to anything in this talk. All right, so today I'm going to kind of tell you two different stories that are hopefully related, but kind of involve two different data sets and two different approaches. The first part is going to be about how we can understand the trajectories of normal adolescent brain development and how that relates to risk for psychopathology. That story is work that we've done on the ABCD data set, some of which those data were collected here at the University of Maryland. And then the second part of my talk is going to be kind of starting from clinical work and understanding kind of a clinical population. How can we sort of work backwards into understanding differences in the brain and get some information that might be useful for us in understanding people's trajectories. All right, so starting with normal adolescent brain development. So the brain matures from early in the prenatal period into childhood, adolescence, and young adulthood. There are a number of processes that occur during brain development, starting with neurogenesis, so formation of neurons. Those neurons then migrate. That occurs primarily during the prenatal period. Then we have formation of glial cells, synapses, and then as we start to get later into childhood and early adolescence, we see myelination of the axons forming and strengthening, and then synaptic pruning. So we think of this as, we're taking away the inefficient and unnecessary synapses. The brain is becoming this really kind of efficient machine to answer the questions we want to answer. And if we look at this, all of these processes together, these top two processes that occur primarily during later in childhood into adolescence come together to form something we would call cortical thinning. So when we look at MRIs of children and adolescents, we see that the gray matter thins. And this is normal and expected during the adolescent period. And it's a result of both the loss of the synapses, but also expansion of white matter so that the gray matter is thinner. Now, if we overlay this with the onset of psychiatric disease, what we see is that our sort of serious mental illness or our major kind of adult diseases that cause a significant disability and burden have a kind of onset that really overlaps. I'm going to do it this way. Okay. Have an onset that overlaps with this time period of cortical thinning. And a number of studies have shown that there are some alterations or changes in cortical thinning that are associated with diseases like depression and psychosis. In particular, I'm interested in early psychosis and psychosis risk. This is just an example. So this is a meta-analysis that I was not involved in. But in this, they are looking at regions of the brain where there is thinner cortex across different stages of psychotic illness. So in purple, you see regions where the cortex is thinner in people who meet criteria for the clinical high risk for psychosis syndrome. And I'll talk a little bit about that patient population later in the talk. Some percentage, about 20% of those people go on to develop a psychotic disorder. And so in green, you see the first episode psychosis population. And again, you see regions of the brain where we have thinning cortex, and that is some in the prefrontal cortex as well as in the insula. And then in blue, we see areas of thinning related to chronic psychosis. And so we look at this and we think, are these changes in the brain related to risk for the disease? Maybe these are sort of underlying vulnerabilities that are present early. It also could be that these changes are related to disease progression. So having psychosis is probably not good for your neurons, right? We have this excess of glutamate. We have kind of, a number of neurobiological changes that are likely to have knock-on effects on the brain itself. And so perhaps these changes we're seeing are actually correlational. They're not causal. And then we also think about, we have treatment effects. So people take antipsychotics and, get other kinds of treatment. Perhaps some of these changes we see in the brain could be related to those. And so many of our studies have been kind of these case control or longitudinal studies where we're following people who already have a disease, and we're trying to draw conclusions from that, which is informative, but doesn't help us with these kind of causal questions. So from there, it has kind of led into this question of really trying to understand, what does normal adolescent brain development look like? What is the normal pattern of cortical thinning across the brain? Because we need to know that to be able to then answer questions about how does that diverge or change in people who develop disease. This study here is one of the kind of best and most well-known studies of cortical thinning during childhood and adolescence. This is a sample out of the NIH. This is 13 kids who were scanned 4 times over the course of childhood and adolescence. You can see in this figure, I'm just going to, the regions that have the warmer colors, giving up on that. The regions that have the warmer colors are thicker, and then as the colors become cooler, these are regions that are thinning. So this is 52 MRI scans in here. And then if you break apart the brain and look at the trajectories of different regions of the brain, you can see there's some differences by region, but that broadly these seem to follow a linear pattern, so that cortical thinning just kind of occurs in this straight line. And so this is useful and informative, but quite a small sample. And as we have learned over the past, 20 or 30 years of neuroimaging, is that we actually need much larger samples to be able to rigorously answer these questions. Additionally, younger kids, as probably is surprising to no one here, you don't need to be a child psychiatrist to know this, aren't great at staying still in the MRI scanner. And the kind of degree to which you're able to maintain stillness in the scanner is directly related to the quality of the data you're getting. And so if we are including poor quality data from younger children, we are inherently biasing our results and our understanding of cortical thinning. And so this is a paper that our group published earlier this year, showing that we have kind of systematic bias if we include these poor quality scans. So we need a bigger and better sample. Now, I've already alluded to the bigger and better sample, and I think many people here are familiar with that. But just briefly to review, the Adolescent Brain Cognitive Development Study. So this is the largest long-term study of brain development and child health in the United States. 11,875 children were enrolled at ages 9 to 10 in a 10-year longitudinal study. I'm not a graphic designer, clearly, but I'm a psychiatrist. I'm going to focus my talk on just a subset of the data that were collected in this study. There are lots and lots of really cool things that they collected. But every 2 years, they brought people in to do an MRI scan. And every year, they collected parent and/or self-report of psychiatric symptoms. And so for this talk, we're going to focus on the data from baseline through year 4 follow-up. So these are people who were kids who are 9 to 10 at the start, 13 to 14 at the end of this data set that I'm describing, but there will be more data in the future, and more data has already been collected. So we wanted to answer this question of what is the normal pattern of cortical thinning so that we can then understand how it diverges. And this is work that was done initially when I was a postdoc working with Josh Rothman and have continued as a faculty member. This figure is just showing you how many MRI scans were available when we did this analysis. And to note this SHN less than 63 as we limited it to data that had what we considered to be adequate quality. So we then ended up with a sample size of about 2,200 children who had 3 time points of good quality data, which is like an amazing sample size in neuroimaging these days, and really what we felt like we needed to be able to appropriately answer this question. And then we looked at cortical thickness. So I mentioned cortical thickness before. How do we measure that in a large sample of MRI scans? 6,600 MRI scans. Is some poor undergrad sitting there tracing the brain like I used to do when I was a Hopkins undergrad? No, we can do this in an automated fashion. So this picture here is a slice of the brain. The red line represents the boundary between the CSF peel matter side and the gray matter. The yellow line represents the boundary between the gray matter and the white matter. And then we're looking at the thickness of that gray stripe around the brain. And then we subdivide the brain or parcellate the brain into regions based on their kind of functional and anatomic information. So we used what's called the Desiccan-Kiliani Atlas. It's 68 regions across the cortical surface. And then we asked several questions. First of all, what was the shape of cortical thinning? Do we see this linear pattern of cortical thinning? Is it just a straight downward line? Preview, that's not a sort of pattern we typically see in biology, but that was what has been kind of understood up to this point. And then did the pattern vary across the brain? So typically we think that there is kind of some spatial relationship to how the brain develops. We often talk about teenagers not having a developed prefrontal cortex, and that kind of more primitive areas might develop earlier, and more kind of multimodal or sophisticated areas developing later in the brain. So what we found was that using this large sample size that has 3 time points of good quality data, that large portions of the brain did have a linear pattern. The regions that are shown here in green are regions that we found this linear trajectory of cortical thinning. The more, the darker green you see were places that had a smaller p-value. And this here is just a representative graph of the thinning in this one particular region. This is a hex plot. So the darkness of the hexagon represents a larger number of people with that data point. And we have age on the x-axis and cortical thickness on the y-axis. And so you can see this region in the inferior parietal lobule has just kind of this downward trajectory over this period of child and adolescent development. However, we also found quadratic patterns of thinning, and I'm going to require everyone to think back to calculus, which is painful for many of us. But what we found were that these regions that are colored in red have a quadratic positive pattern of cortical thinning. And so that is to say that there is kind of this acceleration phase that then flattens out. These regions are primarily in the primary visual cortex, and we would think that these would be regions that develop very early. And so that these regions are probably actually sort of finishing cortical thinning when you're approaching this kind of 13, 14, 15 age range. And then we also found regions that had a quadratic negative pattern of cortical thinning. And you'll notice that these regions are primarily anteriorly in the kind of medial prefrontal cortex. And so these are regions where we have sort of initially a flat pattern, and then it starts to accelerate. And so we think that these are regions where cortical thinning is just starting to pick up at this time frame. And so these are probably regions that are going to continue to undergo kind of more significant maturation. All right. So back to the calculus. If we put these three patterns together, what this ends up looking like is a cubic function. So that kind of thinning is flat, and then it starts to enter this acceleration phase as you get into the, as you get into the quadratic negative, it starts to accelerate, and then you have this linear pattern in the middle. And then it starts to flatten out as you get to this quadratic positive pattern. So this is kind of the first time that we've been able to represent a function like this in the brain, and gives us some really interesting information about how the brain might be developing. Now, so what? Who cares? Calculus in the brain. Well, we can put this a little bit into context. One of the ways that we wanted to put this into context is to think about kind of overlapping function. So this is a map of what's called the sensory motor association axis. So this was developed by Ted Satterthwaite's group at the University of Pennsylvania, where they combined a whole bunch of different maps of the brain to develop an atlas that describes kind of how the brain goes from primary sensory processing regions to multimodal association areas. And so in the lighter colors here are sensory motor regions. The darker blue are multimodal association areas that kind of integrate lots of information, do more executive function, kind of much more higher order functions. And if we look at this map and we compare it to the map of the quadratic function related to age, what we see is a lot of similarities. So regions that are in light yellow on the map on the left are very overlapping with regions that are in the light blue on the map on the right. So these are regions that had that quadratic positive function that we thought were thinning early, seem to map onto primary sensory regions based on the sensory motor association axis. Regions that are thinning later that have this quadratic negative seem to be mapping onto more multimodal association areas. We then took these maps and we looked at the relationship between these rankings, so kind of how quadratic they were, positive or negative, and how sensory motor they were. So the sensory motor axis is shown on the x-axis of this graph, and the age squared beta is on the y-axis. And so what you see is regions that have more quadratic positive thinning are more sensory motor. Regions that are more quadratic negative are more likely to be in the multimodal association areas. So this gives us some suggestion that this is kind of following this functional pattern. Now we're going to bring it to the so what psychiatry question. So are these brain changes in any way related to symptoms? Are changes in the brain driving symptoms? Are symptoms driving changes in the brain? And we can actually use this data set to start to answer some of those questions. So we tested 3 different relationships between symptoms and the brain. One was a delayed pattern. So it might be that if you are a kid who has depression, that is changing how your brain develops. And that, as a child psychiatrist makes sense, right? And so we can test, do symptoms predict later changes in the brain? There might be a concurrent pattern where you have changes in the brain, changes in symptoms at the exact same time. Maybe there's a third factor that's driving both of these, or they're occurring simultaneously, and one is not sort of predictive of the other. And then finally, we have a predictive pattern where we're saying differences in the brain, differences in brain development, then predict later onset of symptoms. And so we tested each of these three models, and we tested them using a dimensional parent report of psychopathology, so the child behavior checklist. This is a 113-question parent report of psychiatric symptoms, and the scores are normed and T-scores based on the sex and age. Anything that's greater than 60 represents clinically significant symptoms. And then there's a total score, internalizing symptoms and externalizing symptoms. So what I've graphed here is just the percentage of kids in the study who had clinically significant symptoms on these three scales. So the purple is the total score. So somewhere around 10% of kids meet clinically significant symptoms in the ABCD study. The green are the externalizing symptoms. As you expect, the percentage of kids with clinically significant externalizing symptoms goes down as we go on to the study, versus the internalizing as sort of flattening out, at least at this time point in the study. We'd probably expect that to kind of pick up as they go further into adolescence. So we tested for these three models, and the only one that we found was significant was this predictive model. So we found that differences in cortical thickness predicted later onset of symptoms. And specifically within these regions, so regions of the visual cortex, posterior cingulate, primary motor, and some parietal regions, the thickness in those regions predicted later onset of symptoms. And if we break that down into internalizing and externalizing symptoms, we saw a lot more predictive power of internalizing symptoms across much of the posterior cortex. So kind of what does that mean? How do we interpret these findings? One of the ways we went about doing that was to do what we're calling a breakpoint analysis. So we're looking at this quadratic function, and we're figuring out where is the bend in the function? When does this thinning sort of start to flatten out in this region that has this quadratic positive pattern? And we can measure that bend in years. So what I'm showing here on this graph, this is cortical thickness for the kids who had the lowest 1/3 of CBCL scores, so the kind of most healthy part of this sample. And then if we look at the kids who had clinically significant scores on the CBCL, what we see is there's actually a later breakpoint. So these kids are sort of flattening out at a later time point. So the first graph is at 12 years or 11.99 years, and the second graph is at 12.19 years that kind of breakpoint occurs. So not a huge difference, but kind of some starting to understand that there's maybe a different trajectory of cortical thinning depending on kind of which group of kids you're in, that kind of kids who are developing more symptoms seem to have a later pattern of maturation. All right, so to wrap up this first part, cortical thinning seems to follow a cubic pattern that has this early inflection point where thinning starts to accelerate, and then in a later inflection point where thinning starts to decelerate. Sensory motor regions seem to be completing thinning during early adolescence, whereas multimodal association cortex is just beginning to thin at this time. And that this variability in cortical thinning patterns predicts onset of symptoms 2 to 4 years later. In the future, we're going to add more time points because, of course, there are more data. This project actually came about because we were looking at the effects of the COVID lockdown on the trajectories of brain development in ABCD, since COVID occurred in the midst of ABCD data collection. When we were doing that, we started to see, the pattern of cortical thinning is not linear as we expected. We're seeing this inflection point. We thought at first it was related to COVID, and it turned out it was not. So it has turned into this project. But we're going to go back to that at some point. All right, now I'm gonna switch gears and we're gonna kind of go to the clinical side of things. So we're gonna talk about some clinical data and diagnosis and clinical high risk for psychosis. So some of you may be familiar with the clinical high risk for psychosis construct, but we'll start with a case study. So Mike is a 16-year-old sophomore in high school. He's always been a little bit quiet. He's a B student. He does extracurriculars. He's got close friends. He has a grandmother who has schizophrenia. And his parents referred him to be evaluated by some people in the kind of early psychosis world because he's had some changes. So he's not hanging out with his friends and his family as much. His grades have started to drop. He sees shadows out of the corner of his eye and he starts hearing his name called. Sometimes he'll kind of ask, like, wait, who is there? Did someone call me? When he's by himself. He deleted all of his social media accounts because he was really worried about internet security. We all know that's a red flag for any teenager. And he was at a concert lately, at a big stadium concert, and he was really convinced that the artist was making reference to him personally, that like some of those lyrics were really just about him. He's talking to his friends about it. Everyone was like, oh, what's going on with you? So this is kind of a case study, a composite example of someone who might meet criteria for the clinical high risk for psychosis phenotype or syndrome. So it's based on 3 decades of research studying adolescents and young adults who identify people in the psychosis prodrome. It's diagnosed based on a structured clinical interview, and individuals typically present what we would call attenuated psychotic symptoms. So they might hear voices or sounds, have paranoid thoughts, disorganized thinking, or changes in their cognition. They typically maintain insight regarding these experiences and they can reality test. So if you ask them, what do you think was going on? Much of the time they can say, I know that sounds crazy. I know this doesn't make any sense, but I'm still having this experience. And depending on kind of who you ask, but approximately 20% of them go on to develop a psychotic disorder within 2 years. Here at the University of Maryland, we have the Strive for Wellness Clinic, and Gloria is here as the medical director of the program, which evaluates and treats these patients. I will, in this, be talking about some work I did at Mass General on this, but we actually know that clinical high risk for psychosis syndrome is much more common than many of us think in general mental health settings. So this is a meta-analysis that we published this past year, where we looked at studies where they went into a general population, not a psychosis specialty clinic, but in a general outpatient mental health setting, counseling center, psychiatrist's office, and screened everybody. And I will note that this sample is primarily adolescents, so the mean age in the sample is 14. But they screen, this is a meta-analysis where we're combining samples of, in total, 14,000 young people who were screened, and looking at the rate of the prevalence of clinical high risk in this patient population, so in general mental health. And when we combine, so each one of these lines is a study where they did this screening using gold standard clinical interviewing, and each square is the prevalence in that sample. On each side, you can see the confidence interval for each sample. And then here in the bottom, that black diamond shows the overall estimate of the prevalence. And so if you screen adolescents, about 26% of them meet criteria for clinical high risk based on doing these gold standard interviews. So it's actually quite common. And it's associated with a lot of comorbidity. So if we think about people who kind of are initially assessed for clinical high risk, nearly all of them, so about 80%, have some psychiatric comorbidity, often anxiety disorders, depression. And then if we follow these people over time, about 20% go on to a psychotic disorder. Many, about half, close to half, have anxiety disorders, depression. About 80% of these kids at 2-year follow-up have at least one psychiatric diagnosis, even if it's not a psychotic disorder. And only about 20% have sort of fully remitted and don't meet criteria for a DSM diagnosis. So much of the research in clinical high risk has focused on this conversion to psychosis, and that's kind of how this field came about. But I'm also personally very interested in these other outcomes and understanding how we might predict these other outcomes or understand kind of who's on what trajectory. So this is some work that I did with Cheryl Fu at Mass General, where we kind of combined our clinical data that was collected from our clinical high-risk program there called RESET. So we collected a lot of data when people were initially referred, including a report of symptoms at that psychotic symptoms at that time, clinical data, functioning level. And then when people came into the clinic, they completed a large battery of self-report measures, similar to what we do now at Strive. And then in this sample, 65 people got a diagnostic clinical interview. And then from there, 38 got what we would call the gold standard, the SIPS interview to assess for clinical high risk for psychosis. That was followed by a feedback appointment where we gave people information about kind of what we think is diagnostically happening for them, what treatment we might recommend, whether or not they met CHR criteria. And so what we found, similar to many other people, is that we had lots of comorbidity. So in dark orange here are people who were referred to the clinic for evaluation. Those in the lighter orange are those who met clinical high-risk criteria. So about 80% or 70% of those who were referred had 2 or more diagnoses, primarily mood and anxiety disorders, but we also saw a fairly high rate of OCD, PTSD, and autism was also well represented in this sample. And then we look at dimensional psychopathology, so people who completed this self-report battery, kind of what symptoms were they reporting on the self-report measures. We found that they had high rates, both people who didn't meet criteria for clinical high risk had high rates of depression. Emotion reactivity is a scale that measures kind of your degree of emotion reactivity. So higher means you are more reactive, sometimes related to kind of borderline personality traits, and then high rates of perceived stress. These dotted lines here are the kind of cutoff for having moderate symptoms. One thing that we found that was really interesting was that those who met clinical high risk for psychosis criteria reported more hours awake, so spending less time asleep, which is something we've become really interested in. And I think as a field have become really interested in this idea that sleep is a major risk factor and a really important thing when we're thinking about the onset of psychotic symptoms. And I should have included in here, but Stephanie Korinek, who's a psychologist at UMBC, and Gloria and other folks in Strive are starting to pilot specifically a group intervention for to improve sleep based on some of these findings of poor sleep. So one other thing that I have started to appreciate, and I'm really interested in this idea that not only do they have these people have high rates of symptoms, but there's a lot of fluctuation in symptoms, kind of from assessment to assessment. These data here are positive psychotic symptoms, and you can see just each line as an individual person. This is 100 people who are randomly selected from a large sample of people with clinical high risk called the AMP-SCID study. And we also see a lot of fluctuation in anxiety and depression symptoms in this patient population. So this has been something I've gotten really interested in, this idea of like, not only do these people have high rates of comorbidity, but there are also just all these kind of fluctuating symptoms that are happening. And how do we understand this? Does this have kind of clinical meaning, or does it help us to predict anything? So it has led to a couple of questions that I'm working on trying to get a handle on. So what causes these symptom fluctuations in clinical high risk? And then can we predict kind of what bucket people are going to end up in by looking at the relationship between symptoms, behavior, and brain networks? So I'm going to briefly explain how we look at brain networks. So this involves using resting state connectivity. So this is a functional MRI scan where people are just laying in the scanner with their eyes open at rest. We know that 90% of the brain's metabolism is active at rest. And so actually when we do a task in the scanner, we're only measuring really minute changes in blood flow. We can get the kind of broad brain patterns of activation, and we extract a signal, so a time course from a region that we would call a seed region. So we'll pick one spot in the brain, look at the time course of activation, and then we look for other regions in the brain where we see a correlation of the time course. When we do this over time, we can start to see these patterns emerge, that there are distinct networks within the brain that are connected to each other. We know that we infer connection based on this correlation of time course, because we know that things that fire together wire together. If we see a correlation and pattern of activation, that those regions are talking to each other. And these patterns, these networks are stable over time. We can reproduce them both within individuals and between individuals. And we can look at the relationship between symptom variability and these different networks. So you might imagine that we have a couple people who meet clinical high risk for psychosis criteria, and we might be tracking their symptoms over time. So we're tracking anxiety. This is fake data. I made it up in PowerPoint. Anxiety, depression, psychosis. And we can kind of follow that over time and look at these fluctuations. And then what I'm interested in is understanding kind of what is predicting these fluctuations. Does that have any meaning? And so to do that, I'm using a paradigm that a collaborator, Chuck Lynch, who's a neuroscientist at Cornell, has done, where he did something similar in people with depression. So he brought people with major depression into the lab at Cornell every week. This one person was followed like over a year. So here you see in that black line, this is tracking this person, individual's symptoms of anhedonia. All of those black dots are when they got MRI scans. So they got resting state MRI scans like I just showed. And using a really advanced resting state sequence, he can actually develop an individual level map of the different networks in that person's brain in a way that is reliable and really is kind of a signature, a fingerprint of their brain networks. And what he found is that the salience network, which is a part of a network in the brain that's involved in detecting things that are emotionally salient, which you might imagine could be quite important in depression, that two nodes in that network were specifically related to symptoms of anhedonia in this individual person. So the connection between the anterior cingulate and the nucleus accumbens, which is a region of the brain involved in reward processing, was specifically related to anhedonia symptoms and actually predicted anhedonia symptoms 1 week later. He then replicated this in a second person who was scanned in a similarly intensive fashion. That person was only scanned for about 8 weeks, but again, found a similar pattern in these same connections. So we are using this technique to kind of examine and try to understand some of these different symptom fluctuations within clinical high risk. So Stephanie Korinek is working with me on this as well. She's a psychologist at UMBC. So we are bringing people in every week and measuring their symptoms. We are getting some objective measures of their behavior. So they complete an open-ended interview that's video recorded with one of our research assistants. From there, we can extract affect. So the intensity and types of expressions that they make. We can get speech and language data from that video. And then we're also collecting measures of sleep and physical activity. So our participants are wearing a little watch that measures how much they move around, and we can get sleep and physical activity from there. And then the hope is to pull all of this together and to see if we can understand this relationship between behavior, self-reportive symptoms, and objective and the resting state networks. So this is something we have just started. We had our first scan of a control yesterday, so it is very early on, but I'm really excited about this project. And the whole goal of this is really to try to understand trajectories. So we're going to be following these people with scans for about 6 months, but we'll be longitudinally following them for 2 years if the NIH gives me money to do this, so that we can start to understand, hopefully get more understanding of these trajectories and the heterogeneity here. All right, so future directions of this, as I mentioned, kind of this longitudinal clinical outcome prediction. One of the things that, of course, when we look at networks, then can we perturb networks or fix networks in some way? So can we modulate these circuits if we find differences, either TMS or low intensity focused ultrasound? I mentioned the AMP schizophrenia data set. I'm also looking at these networks and symptoms in there. And so overall, lots of exciting things to do, lots of wonderful people who've been working on this with me. So these are some of my many collaborators and funding sources. And with that, I'll take questions.
We have the microphone in here, so if you have a question, just raise your hand and shout it out, and then we can go into the chat. I have one quick question.
Yeah.
So and this is coming from the perspective as a geriatric psychiatrist.
Sure.
How long have they followed the ABCD people, and how long will they be followed? It could be very powerful data. The reason I ask is there's a very famous study in geriatric psychiatry called the Nun study, where these nuns who were like 18 years old. And based on their writings at 18 years old, you can predict who got dementia. So I just think that, I don't know how old are the oldest folks?
So yeah, Gloria's involved in the study. So
18
right now. So
are they looking at funding like 5 or 10 year fellow beyond that?
Yeah, so I think so a lot has been invested in the ABCD study, and it's a predictable time. So we'll see what happens. But at the site visit, they've talked about, preparing for, there may be opportunities to invite people to participate longer.
Retention is challenging.
Yeah.
They're in college now, so they're starting to,
Yeah. So I think it was planned for a 10-year follow-up, but who knows? And, some of the clinical high risk data that I presented, typically people are followed for 2 to 3 years, but there's a group that is kind of actually going back to find some of these people who are in some of these studies like 15 or 20 years later, and they're following them on social media. And Kristen Cadenhead out in California is working on this to find, especially the people who dropped out, because you might imagine that typically the sickest people drop out, and we're probably missing a lot of kind of conversion to psychosis because of that. Yeah, it's an important question and a real challenge for these studies. Yeah.
It's such an interesting talk. Thank you, Jackie.
Yeah.
Is there much known about sex differences in either time or rate of approval?
Yeah, so that's so the kind of broad belief is that boys mature later. We have to, and that's actually been a debate between me and Josh about whether or not we should dig into that for this paper. We're hopefully wrapping it up to submit. But it is a really interesting question and probably a lot to sort of unpack there. So we haven't dug into it, but I think broadly we think that the maturation patterns are different, as you would expect. Yeah.
It's been in the chat.
It would just be in the group chat. Is that correct?
Yeah.
Okay. Oh, there we go.
Yes.
A general question. Okay. Yes, trauma. So the question is, the slide on clinical outcomes for CHR didn't see anything regarding trauma disorders in the first episode clinic and inpatient setting. We often see significant overlap between attenuated psychotic symptoms and dissociative spectrum complex trauma symptoms. If that's been explored in CHR. Yes, great question. So we know that trauma is one of the biggest risk factors for onset of psychotic disorders. It's a tricky thing within the sort of overlap between dissociative PTSD spectrum symptoms and psychosis is something that's really challenging to try to disentangle. And we will often sort of say, okay, if we can attribute this symptom to trauma that we would say it's trauma related. And if we can't, then maybe it's more psychosis. But I think that that's probably a far overly simplistic way of approaching it. I have an MD PhD student that I mentor back at Harvard who's looking at the psychotic-like experiences in the ABCD study and kind of fluctuations in those over the course of the study. And one of the best predictors, the two best predictors she's found for people having high levels of those symptoms are trauma and bullying, traumatic life experiences and bullying. So I think that that's a huge piece of this that we don't fully appreciate and is, again, gets back to our constant chicken or egg question in psychiatry that is hard to answer. Yeah.
I know that a lot of times in courses we talk about like negative symptoms as well, and so I'm curious if, in addition to seeing areas that are more activated with certain networks, if you're seeing places that are a little more dark and tend to be like more inhibited in terms of that activity.
Yeah, so when we talk about the resting state connectivity, we're not necessarily looking at it's not the sort of beautiful blotches of activation or deactivation. It's instead the strength of the connections. And so absolutely people with clinical high risk for psychosis often have similar levels of early negative symptoms. And that's probably a really important piece of all of this. Negative symptoms are predictive of long-term functioning, are some of our hardest symptoms to treat. So it's something we really need to be thinking about. For this study, one of the reasons we're doing the video recording is we can actually measure affective blunting, and we can measure kind of reduced cadence of speech, and we can measure in an objective way some of those pieces because that is really important. Yeah, great question. Dave Gorlick asked about psychoactive substance use related to trajectory or association with psychiatric disorders. Yeah, so of course, smoking pot is bad for your brain and makes you more vulnerable to psychosis. There has been less done on the relationship of marijuana and clinical high risk than I would expect. We do have a study at Mass General where we're starting to do a family-based intervention to teach parents how to do motivational interviewing to get their kids to stop smoking pot within the clinical high risk population. But, I think it's an important question. Someone had recently asked me about thinking about like the polygenic risk for substance use and how that might relate to this as well. And to my knowledge, nobody has done anything with that. But what I often counsel people clinically is like, if you're in this clinical high risk group, you are probably the most vulnerable, and it is the most important for you to stop using psychoactive substances more than others. But yeah.
And just sort of a follow-up, because we talk about the effect of things on brain volume, and we know that trauma is associated with different brain volumes and subcortical structures.
Yeah.
So kind of a two-prong is, how are you able at this point to make any comment about the relationship of that with the cortical structures that those subcortical structures serve? And are you tracking substance use in your population as you go to see if that's a variable on how things thin and don't?
Yeah, good question. So for this particular study, for the ABCD analysis, we have yet to look at the subcortical structures. So we expect volume of subcortical structures typically kind of continues to expand as you kind of go through into adolescence. But then as people develop, yes, things like PTSD, psychosis, we see particularly smaller hippocampal volumes over time. And people have done some really nice studies looking at twins and things like that for that. We wanted to focus on the cortical development specifically for this, so I don't have an answer on that. The ABCD study actually is a partnership between the NIMH and NIDA. And so the time period and many of the measures were specifically chosen to track substance use. So trying to get kids before they started using substances and to kind of use that to be able to develop predictive models. With the data we have right now, there's very low reports of substance use so far. In the first 4 years of the sample, that's reports, not necessarily use. And so, some of that is probably related to, reporting bias. I know that they are collecting urine samples to do some tox screens, but obviously that doesn't capture everything. So, we'll see how the data comes out. And then, of course, we have a significant dropout rate from the study. And you would expect that, families who are struggling more, with things like substance use are not going to be showing up for their appointment for the ABCD study. So I think it's a challenge and it's a really important question. Yeah.
They also collected little baby teeth from the kids.
Yes.
For in utero drug exposure.
Yeah.
Just, as we're talking about this, given how the huge wave of marijuana.
Yes.
That is, I can't walk from the main hospital to the 701 building without smelling a skunk anymore.
Yes. Yes.
And so with that, I'm just kind of wondering how we're going to be able to look at data on such a changing substrate. It really seems very challenging.
Yeah. No, I think that's a great question. And I will be curious to know. I don't have the numbers off the top of my head. I know that at least up to the year 4 data, there's maybe a chunk who've at least reported trying alcohol, but very few report trying marijuana. Now, I agree with you that's probably not accurate. Yeah.
I remember how many times I told about such things when I was.
Yes. And especially a challenge of if you report these things, it might mean that you have to stay longer and answer more questions. So, yeah. Other questions?
Thank you.